Okay, in this video, what I'd like to do is a couple of things. The first is to show you how to properly design primers, and then the second one is to show you how a PCR reaction takes place, to show you the products that are formed in the first 3 or 4 cycles. And the reason I want to show you that is because I want to show you how primers and the restriction sites that you add to primers can be incorporated into the PCR product. So that, as we explained in the prior video, if you need to do your cloning via PCR because you need to add specific restriction sites to your PCR product so that you can create compatible cut sites that match with the vector, then you need the primers to be incorporated into the PCR product. And so I want to show you how that works. So what you're looking at here at the top of this screen is a gene sequence that I pulled off the web. It's not GFP, it's another gene. And I've highlighted in yellow the start codon and in green the stop codon. And I've also put ellipses where the intervening actual gene sequence is, and I've also included some of the flanking sequence. So the flanking sequence would be this sequence just before or upstream of the start codon and just downstream of the stop codon. And the reason that we need that flanking sequence there, we need to know what it is because we need to put our primers in, we need to make our primers so that they anneal to that flanking sequence. Because what we want to do is copy the entire gene. And so we need to have our primers anneal outside of the gene, both upstream and downstream of it, so that we can, so that when we make the copies via PCR, the entire gene is copied. So if you look at a gene sequence like this, and these sequences are found in many databases on the web, and you are able to locate the start and stop codons, then you can design primers. And I want you to notice that this sequence only contains the sequence for one of the 2 strands. And it's the sequence that actually has the coding sequence in it. The reason I know that is because it has a start codon and a stop codon. So this sequence here, ATG, TTC, AGT, etc., is the actual sequence for the gene. Now the other strand isn't shown here, but we can write some of it out. I don't have time to write it all out, or maybe I will and just edit the video so that it's written out quickly. Okay, I've written the other strand, the sequence for the other strand out. I've skipped some of the bases here in the middle. These I didn't write out. So here's the 3 bases that are opposite the start codon, and here are the 3 bases that are opposite the stop codon. And then what I have over here and here are the flanking sequences. Now, these strands are given different names depending on whether they contain the coding sequence, like this top one does, or the other, what we call antisense sequence. So this top strand is given the name the sense strand or the coding strand, and this bottom strand is given the name antisense and non-coding strand. And don't mix up the word antisense with nonsense. It's not the nonsense strand, because nonsense would mean that when you refer to nonsense, when you talk about a DNA sequence, you're talking about a stop codon that stops translation. This is the antisense strand, and this is the sense strand. And because I know this is a sense strand, and again I know that is the sense strand because it has the start codon ATG, which is the universal sequence for start codons, then I know that this gene is that this end of the DNA is the 5' end, and this end down here is the 3' end. And that would mean then, because DNA is anti-parallel, that this end would be the 3' end, and this complementary this end would be the 5' end. So this strand, the sense strand, we would say is complementary to the antisense strand, and it's also anti-parallel to the antisense strand. Okay, now let's think about how we're going to design primers that will encompass this gene from the start codon to the stop codon. So we're going to have to design primers that anneal to the flanking sequences. And I want you to recognize a couple things. First of all, if you design a primer that anneals to DNA, it's going to have to anneal in an anti-parallel fashion, just like these two strands stick to each other in an anti-parallel fashion. In other words, if I have a strand of DNA here where the 5' end is here and the 3' end is here, and I develop a primer that anneals here, it would be I would have to design it such that the 5' end is on this side and the 3' end is on this side, so that these two strands are anti-parallel to each other. So that's the first thing I want you to recognize. The second thing I want you to recognize is that when we copy DNA, the DNA polymerase attaches to the 3' end of a primer. So I have a piece of DNA here, and I have a primer here, and a primer here, and this is the 5' end, and this is the 3' end. That would make this the 3' end of the primer, and that would be the 5' end of the primer. Same thing over here. If I were to copy this using DNA polymerase, so let's add the DNA polymerase. Let's add dNTPs, which are the nucleotides, a mixture of A, T, C, and G. These are the building blocks. And I'll add A, T, C, and G. And I'll add a buffer to keep the pH at the proper level and the salt concentration at the correct level, ion concentration at the correct level, so that DNA polymerase works. So if I have a mixture of primer, template DNA, which is the DNA that I'm going to be copying, DNA polymerase, dNTPs, and buffer, then the primers will anneal to the template DNA in an anti-parallel fashion, and then DNA polymerase will attach to the 3' end of the primers, and it will copy in this direction. It will read the template DNA in the 3' to 5' direction, but it will copy new DNA in the 5' to 3' direction. So it reads in one direction, 3' to 5', but copies or adds new DNA in the 5' to 3' direction. So DNA polymerase would attach here at the 3' end of the primers, and it would start producing new DNA going in this direction. It would start adding dNTPs. And of course, if this polymerase started moving down the track, once it got to this primer right here, it would fall off because it couldn't go any further. But this DNA polymerase, depending on how far this DNA strand goes in this direction, could theoretically continue on in that direction for a long distance. So if this were my gene right here, where would I want my primer to attach? Well, if you think about it, if I made a primer that attached right here, the DNA polymerase would synthesize DNA off into that direction, and my gene, which is right here in the middle, wouldn't get copied. Instead, what I want to do is make sure that my primer attaches at this end so that the gene is copied. Because if I attach my primer down here, the DNA polymerase will attach to the 3' end of the primer, and it will synthesize new DNA in this direction, which will include my gene, which is right here. And so I want you to recognize that when we design primers, we have to take into account the 5' and 3' directions of the primers, and also the direction that DNA polymerase will travel. So in doing PCR and making a copy of a gene, we need 2 primers. And the primers are given names. They're called the forward and the reverse primer. And the forward primer is going to sit somewhere in the upstream flanking region, and the reverse primer is going to sit somewhere in the downstream flanking region. And so when I design a primer, the question will be, let's start with the forward primer. When I design the forward primer, the question is, what sequence should I make this primer? Well, I need to make a sequence that is complementary to one of these two strands. But which strand should I make it complementary to? Well, let's take a look at the sense and antisense strand and see. Let's pretend that I make a primer that is complementary to the sense strand. Since the sense strand reads 5' to 3', the primer down here that attaches to it, anneals to it, would read 3' to 5'. And when I add my DNA polymerase and my nucleotides, DNA polymerase would actually copy the DNA in that direction. And so if I make a primer that anneals to my sense strand, I'm not actually going to end up copying my gene, which is not what I want. So I hope you can see that when I make a forward primer, it needs to anneal to the antisense strand. All right. You've read in your manual some of the other requirements for primers, but this is the big one. To make your primer copy the gene, the forward primer needs to anneal to the antisense strand. You read in your book that primers are generally 18 to 30 bases long. If we know the flanking sequence here, we can design a primer that anneals to it and then check and see if it meets all the standards that we want in terms of its melting temperature, in terms of its hairpin loops, and primer dimers that it might form, which we'll talk about in a second. All right, so let's design a sequence that would anneal to this antisense strand. All right, I'm just going to count up from my start codon 18 bases. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. And I'm going to design a primer that would anneal to this antisense strand 18 bases up. So T, G, 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 A, G, G, A, G, C, G, A, C, 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 G, G, G. Okay, there's an 18 base pair primer that would anneal to the antisense strand. Now, if I look up here at my sense strand, I can just do a check and make sure that it's the exact same sequence because it should be. All right, so TGG, TGG, GAG, 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 CGA, CGA, CCC, CCC, GGG, GGG. Okay, so this is a good start for a primer. It's 18 bases long. It anneals perfectly to the antisense strand. So I'm going to say this is my tentative forward primer. And then I'm going to do the same thing with the reverse primer. Now it needs to anneal downstream of the stop codon, but which strand should it anneal to? Well, if I made it so that it annealed to the antisense strand, which has a 5' end here, that would mean this would be the 3' end of the primer, and this would be the 5' end of the primer, since it would attach in an anti-parallel fashion. And so when I added my DNA polymerase, it would bind right here and then copy in that direction. And so my gene, again, wouldn't be copied. It would be going the wrong way. So instead, I need to make sure that my reverse primer anneals to the sense strand. And so I'm going to just count up 18 bases here and design a primer that anneals to the sense strand on this flanking DNA. So T, G, T, T, C, T, G, C, A, T, A, C, C, T, C, C, T, C. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Wow, that was a really good guess. All right, so now I've got a primer, and I've got a forward and a reverse primer. And I need to just keep track of the 5' and 3' ends of these primers, because when I order them from the company so that they can send them to me and I can use them in my PCR, I need to tell them which end is going to be the 5' end and which is going to be the 3' end. So this is the 5', 3' orientation of these primers. Primers are almost universally written in the 5' to 3' direction. So if I talk about if I want to write the sequence of my forward primer down, I would write 5' T G G G A G G A G C G A C C C G G G. When I write my reverse primer down, I'm actually going to have to read it backwards, and so it's going to be C T C C T C C A T A C G T C T T G T. Okay, so this is the proper way to write my forward and reverse primer sequences out, so that someone else, when I send them out to the company, they design the exact right primer that I need. So again, I write them in the 5' to 3' direction. Now before I send them off to the company to order them, I'm going to put their sequences into a primer checker, basically an online tool that will check the primers. And some of the things that I want to check for are melting temperature, which is abbreviated TM, hairpin loop formation, and primer dimer. And I wrote delta G here. Let me explain what this means. Let me cover melting temperature first. When we design primers, when we do the PCR reaction, our primers are going to anneal to the template DNA. So in the primer reaction, we heat our template DNA up. The two strands, the sense and antisense strands, separate from each other. And then we cool the DNA down slowly. And as we do, the primers that are in the solution with the template DNA will anneal to the template DNA. This annealing step is very important, because if the primer doesn't efficiently anneal to the template DNA, we won't get copies of our DNA. And so we need to set a temperature that will optimally anneal the primers to the template DNA. When things are hot, the hydrogen bonds between the DNA strands will break, and the strands will separate. But then as we cool down to a certain temperature, those hydrogen bonds can form. And so we need to determine when we set up the PCR reaction a temperature that will optimally allow both the forward and reverse primer to anneal to the template DNA. Because we're going to tell the machine, after you separate the strands with high temperature, I want you now to cool down to allow the primers to anneal. But what temperature do I cool you down to? Well, the ideal temperature is going to be close to what we call the melting temperature of the primers. The melting temperature of the primers is the temperature at which the primers will anneal to the template DNA. It's also the temperature at which they will come away from the DNA, because if you think about it, for example, 0 degrees Celsius is both the freezing temperature and the melting temperature of water, because anything below 0 degrees will be frozen, anything above 0 degrees will be melted. So for these primers, they have a melting temperature, which is the temperature at which they will anneal, but it's also the temperature at which they will start to come off of the DNA. And that melting temperature is determined by the number of nucleotides that your primer has. The more nucleotides that it has, the longer your primer is, the better it will anneal, so the higher the temperature you will need to remove it. And the more G's and C's you have will also determine how high the melting temperature is. Because remember, between a G and a C, there are 3 hydrogen bonds, whereas between a T and an A, there are only 2. So these hydrogen bonds are harder to break because there's 3 of them. And so a primer that has a lot of G and C, a high GC content, we say, will require a higher temperature, will have a higher melting temperature, will require a higher temperature to remove those primers, and therefore they will stick at a higher temperature as you cool down. Let's say you got them hot enough to separate, and then as you cool down the DNA, well, something with a higher melting temperature will anneal at a higher temperature than something with a low melting with a low melting point. Just like if you had two substances, let's say you had two substances, one had a melting temperature of 17 degrees and the other one had a melting temperature of 36 degrees. If you heated them up to 95 degrees and then started cooling them, if you heated them up to 95 degrees and then started cooling them, this one would become solid first, and then this one would become solid second, right? So, same thing with these primers. The one with the higher melting temperature will anneal at a higher temperature. Okay, I hope I didn't beat that dead horse into the ground. But what we need to make sure is that both our forward and reverse primer have similar melting temperatures, so that when we set the machine, the PCR machine, to the temperature, the annealing temperature that we want, both primers will anneal at that temperature that we set. If they have different melting temperatures, then one primer might be annealed, but the other one wouldn't be. And so we want to make sure that these two primers' melting temperatures are within 2 degrees or so of each other. Ideally, they'd be exactly the same temperature. The other thing you want to make sure of is that these primers are going to want to anneal. Want is a weird word for an inanimate object here, right? But you want to make sure that these primers are going to be are more likely to anneal to the template DNA than they are to themselves or each other. You can imagine, maybe by just chance, it happened that these flanking sequences were somewhat complementary to each other. This reverse flanking sequence and this forward flanking sequence happened to be somewhat complementary to each other, so that when you designed a primer, these primers ended up being somewhat complementary to each other. Well, then when you do the PCR, the primers, instead of annealing to the template DNA, may anneal to each other and form a primer dimer. When they form that primer dimer, they can actually be copied, and so the DNA polymerase would attach to these primers and form new DNA here, and so you'd get something called a primer dimer, and that's not going to contain your gene that you're interested in copying, but it is going to contaminate your product. It's going to contaminate your results. So you don't want these primers to attach to each other. So the computer program will also check to see how likely it is that they attach to each other. So a low delta G means higher, or a lower delta G, because it's all relative, means a higher probability of forming. So if I have a lower delta G for primer dimers, that means I have a higher probability of them forming. This is a measure of free energy, and so it's basically a measure of favorability. And a lower delta G means a higher favorability or a higher probability. And so if I put my primers into a calculator and find that the delta G for primer dimer formation is lower than it is for annealing to the template, then that means, so let's say the delta G for primer-dimer formation is lower than the delta G for annealing to the template, then that would mean that I'm more likely to get primer-dimers than I am the primer annealing to the template properly. Hairpin loops are situations where the primer doesn't anneal to the other primer, but it anneals to itself. So let's say that you had a primer where there were a bunch of A's on one end and you had a bunch of T's on the other end of the primer. Then this primer may actually anneal to itself because it has these sequences that are complementary to each other on itself. And this intervening sequence here would form a little loop. This is called a hairpin loop. And this is also a problem if the delta G for forming a hairpin loop is less than the delta G for annealing to the template. Now, there's always going to be some likelihood that these primers anneal to themselves or anneal to each other, but the more important question is not whether they ever will, but whether they're more likely to do that than they are to anneal to the template. And that's the number you want to make sure. That's the number you want to check. Are they more likely to anneal to the template, which is what you want, than they are to form primer-dimers with each other or hairpin loops with themselves? So once you've checked these things and you have determined that these sequences, the forward and reverse primer, have similar melting temperatures and are not likely to form significant hairpin loops or primer-dimers, then you can go with those primers and you can send them off to the company. What if you run into a problem and their melting temperatures are different? Well, what you can do is you can add or take away bases from the primer you currently have to alter the melting temperature. For example, if I added a couple bases onto the end of this primer, these two G's, I've just added two GCs, two GC bonds. That's going to make this primer anneal better, and so its melting temperature will go up. And so if this melting temperature, let's say, was 55, and this one before I added these two G's was 50, maybe by adding these two G's here, I ended up raising the melting temperature of the primer to 54. Now it's within 2 degrees of the reverse primer. And so if you need to change if your melting temperatures aren't matched up, then you can just add bases until you get a close match between the two melting temperatures. Also, keep in mind, I designed a primer that abutted immediately onto the start codon. In other words, I designed a primer that annealed immediately upstream of the start codon and immediately downstream of the start codon. You don't have to do that. You can design a primer that anneals a little bit more upstream. Like I could have designed a primer that was GATC, GGG, AA, GT, GGT. I could have designed this as my forward primer instead, as long as it was 18 to 30 bases long. And I could design a reverse primer that anneals somewhere a little bit further downstream of the stop codon. You don't have to have it immediately butt up against the start codon and the stop codon, but there's no need. If you can have it immediately butt up against the start and stop codon, that's great, because then you don't have this intervening DNA sequence that you're going to be copying. But it's not going to make a big difference if you scoot up a little bit to produce your primers a little bit further upstream and downstream of the start and stop codons, respectively. It's okay if you copy more than just the gene when you do PCR. This is what we really want to copy, right? This is the gene sequence. But it's okay if we copy a little more than that, including some of this flank. Oh, here's the gene sequence, sorry. But it's okay if we copy a little more than that. It was funny I circled that because I saw this ATG and I thought that was a start codon. I want you to see that some genes will have other start codons in them. There's actually another ATG. So how do you know which ATG is the actual start codon? Well, on the map or on the sequence or in the database, it will you should be able to search, and they'll tell you where the actual start codon is, because there's multiple ATGs here, aren't there? So I knew this was the start codon because I looked at the database, told me where the start codon was. Okay, once I've got my sequences for my forward and reverse primers, and I've checked all of these parameters to make sure that they're within proper range, their values are within the proper range. Now I can do one more thing, and that is I can add the cut sites that I want to have these primers to have. Remember, we said that when we have a donor plasmid that has the gene we're interested in cloning, when there are no cut sites that match the cut sites that we find in the MCS of our receiving vector, then what we have to do is do PCR, where we add cut sites to our primers. And so what we're going to do now is add cut sites to our forward and reverse primers that are compatible with the cut sites that we find in the multiple cloning site. So let's say that the multiple cloning site has a cut site for KPN1 and HinD3. Then what we need to do is add a KPN1 cut site to our forward primer and a HinD3 cut site to our reverse primer. And what we're going to add those is the 5' ends of the primers. So the cut site here, the HinD3 cut site, is going to go at the 5' end of this primer and at the 5', and this is the reverse primer, and the KPN1 site is going to go at the 5' end of our forward primer, which is going to be up here in front of the gene. The reason we add them to the 5' end is because these parts, these cut sites, are not complementary to the DNA, and so they will not anneal. These parts here will anneal, these sequences here will anneal, but when I add the cut sites on this end of the of my primer, that part of the primer will not anneal to my template DNA. And so if you think about where the DNA polymerase attaches, it attaches to the 3' end down here. And so the DNA polymerase will attach to the part of the primer that is annealed to the template. If I put my KPN1 sites on, if I put my cut sites on the 3' end, then what you would see is a primer that looks like this, where this part would anneal to the template DNA, but then the cut site wouldn't anneal. And so the DNA polymerase would attach here, and it wouldn't have anything to copy. And so you can't put your cut sites on the 3' end. You have to put them on the 5' end. So I'm going to look up what the KPN1 and HinD3 sites are, and I'm going to attach I'm going to write those sequences onto my primers, onto my 5' ends, so that they'll now have cut sites on them. Okay, the sequence for KPN1 is GGTACC, and the sequence for HinD3 is AAGCTT. Okay, so when I order my primers, I'm going to send to the company, and I'm going to tell them, I want you to send me a primer that's GGTACC, TGGG, AGG, AGCGA, CCCGGG. That's my forward primer. And my reverse primer will have a sequence AAG, CTT, CTCC, TCC, ATA, CGT, CTTGT. All right. Now, this would be almost perfect, but having a cut site on the very end of the primer reduces the efficiency of the enzymes once you actually cut the DNA. So on the very 5' end, I'm just going to add a couple of A's to put that cut site a little bit inside the primer. Okay, so that's a complete primer then, where I have the complementary. It has 3 parts: the complementary sequence that's complementary to your template DNA, the cut site, which is compatible with the cut sites in the multiple cloning site of your vector that you're putting this DNA into, and then it has a couple extra bases at the end just to improve the efficiency of the digest once you actually digest with the restriction enzyme. So those are the 3 parts that the primer has, and then this would represent a complete primer. Again, the forward primer would have a sequence that starts 5', AA, GGT, ACC, TGGG, etc. The reverse primer would have a sequence of 5', AA, AAG, CTT, CTCC, etc. Those would be complete primers. I would send those off to a company and wait with bated breath for them to come back so that I could start my cloning experiment.